Immunoprecipitation and co-immunoprecipitation. In order to raise antibodies to membrane proteins and other cellular structures that are difficult to purify, mice are often immunized with whole cells or crude cell extracts. Antibodies to the individual molecules are then obtained by using these immunized mice to produce hybridomas, making monoclonal antibodies that bind to the cell type used for immunization. To characterize the molecules identified by the antibodies, cells of the same type are labeled with radioisotopes and dissolved in non-ionic detergents that disrupt cell membranes but do not interfere with antigen-antibody interactions. This allows the labeled protein to be isolated by binding to the antibody in a reaction known as immunoprecipitation. The antibody is usually attached to a solid support, such as the beads that are used in affinity chromatography, or to a protein A. Cells can be labeled in two main ways for immunoprecipitation analysis. All the proteins in a cell can be labeled metabolically by growing the cell in radioactive amino acids that are incorporated into cellular protein. Alternatively, one can label only the cell surface proteins by radioiodination under conditions that prevent iodine from crossing the plasma membrane and labeling proteins inside the cell, or by a reaction that labels only membrane proteins with biotin, a small molecule that is detected readily by labeled avidin, a protein found in egg whites that binds biotin with very high affinity. And that's immunoprecipitation and co-immunoprecipitation.